Hi class, and welcome to your next video on DNA and making proteins. So we've already talked about how DNA is a double helix and its specific structure and how it has nucleotides composed of a phosphate, a 5-carbon sugar, and a nitrogenous base. Now we're going to talk about what exactly does the cell do with that DNA and what is that DNA specifically used for. So remember that DNA, like I just said, is composed of 2 strands and it's made into the double helix. Now, it has those nucleotides, which we have the phosphate, the 5-carbon sugar, which is in DNA is deoxyribose, and then it has a nitrogenous base. Those nitrogenous bases can be cytosine, guanine, adenine, or thymine. And remember that cytosine bonds to guanine and adenine bonds to thymine. And if you recall, there are 3 hydrogen bonds in between cytosine and guanine and 2 bonds between adenine and thymine. So the first thing we want to talk about is DNA replication, because remember, we talked about how DNA replicates itself for both mitosis and meiosis. So we are going to need to replicate DNA on multiple occasions. So DNA replication begins when a section of DNA unzips down the middle and is going to use an enzyme, which we've talked about, that's a type of protein, called DNA helicase. And it exposes the nucleotide bases. So we're going to expose these bases right in here. And then another enzyme called DNA polymerase moves along the 2 strands, pairing complementary bases exposed to the nucleotide. So what that means is DNA helicase comes in, unzips the DNA. So now we have the nucleotide bases there. So if we have an A, a T, a G, another G, and a C, then the polymerase is going to bring in a T to pair with that A, an A to pair with that T, a C to pair with that G, another C to pair with that other G, and so on and so forth, so that we get complementary strands. So up here, we're going to have our original template strands, and down here, where we have the duplicated DNA, we're going to make our complementary strands. So in DNA replication, a complementary strand is formed for each strand of the double helix. So we're going to get 2 complementary strands. So that means we have 2 template strands. And we're going to use that. Now, DNA replication can happen at multiple spots in the DNA at multiple times. It doesn't have to go from one end to the other. It can happen all along the DNA at any part of the DNA at any one time. We just have to unzip that part of the DNA, and we have to have the helicase come in first, and then we have to have the polymerase come in and feed in those nitrogenous bases and nucleotides so that we get that complementary strand. So we're going to use that replication for multiple things, but like I said, we're going to use DNA for more than just to be there and be our instructions. Now, DNA cannot leave our nucleus, but to make proteins, we need something to leave the nucleus because our ribosomes are what make our proteins. So DNA is in the nucleus, just like a recipe for making our proteins. It's just like the recipe is giving instructions for making food, and the DNA gives the instructions for making the proteins. However, since the DNA cannot leave the nucleus, and the ribosomes that make the proteins cannot enter the nucleus, we have a little bit of a problem. And so we've talked about DNA, and we've talked a little bit about RNA. So in order for them to receive these instructions, we must use the molecule RNA, which is ribonucleic acid. So the cell makes a copy of the DNA using a molecule very similar that can leave the nucleus. RNA can leave the nucleus it comes to the DNA, basically makes a copy of the DNA, and it's like a photocopy of that recipe. And then it's going to go out to the ribosome, which is going to help make the protein. So if we recall from our unit on macromolecules, RNA is single-stranded. It has the 5-carbon sugar ribose. Instead of, remember, in DNA, we have deoxyribose. And it does not contain the nitrogenous thymine, but it contains the nitrogenous base uracil. So if we look at DNA versus RNA, we can see that we have this double helix in DNA, but we only have the single helix in RNA. And we can see the base pairing in DNA versus there isn't actually a base connected to each nitrogenous base in RNA. We have the bases A, T, C, and G in DNA, and we only have the bases A, U, C, and G in RNA. So we have several differences between DNA and RNA. There's 3 main differences: the sugar, the nitrogenous base, and the actual shape. So the process of transcription. Transcription first starts when the DNA strands unwind. We have to have part of that unwind. And then RNA polymerase this time comes in to make a complementary RNA strand from the original DNA template strand. Remember when we did DNA replication, we were using DNA polymerase. This time we're going to use RNA polymerase because we want to make a copy of the DNA, but we want to make something that can leave the nucleus, so we need that RNA. So then, since RNA does not have the base thymine, now adenine is going to pair with uracil. So every time we have an A, usually in DNA, we're gonna have a T that pairs with it, and the DNA polymerase would say, okay, let's bring in a T. Well, RNA polymerase says, let's bring in a uracil or a U. So the new RNA strand is called an mRNA, as it can leave the nucleus to be a messenger. So we call this messenger RNA. So if we look at this, what would the complementary base strand be to GGC, TTA, GAC, CTA? Pause the video and write down your answer. Now, the correct answer would be CCG, AAU, CU, GG, AU. So make sure you check your answer to that. Now let's look at this. This is the process of transcription. Now remember I said that this is going to unwind, and this is our DNA right here. Our DNA has unwound, and our RNA polymerase has come in. And this is our new RNA strand, and as we can see, the RNA is binding a little bit to that DNA. So it's a complementary strand, but it's not that exact thing. So as you can see, for every A, we don't have an A, but we have a U. And for every G, we don't have a G, we have a C. So we have that complementary base. We don't have the exact base. So once the mRNA strand is made, it can go out and leave the nucleus, and it leaves through the nuclear pores, and then it can go out to the cytoplasm. Once the messenger RNA reaches the cytoplasm, ribosomes will attach to it. They just come out and they kind of see the mRNA and they kind of come out to it, and they know that they are supposed to be there. And they start to translate it. And they start reading the mRNA and making the proteins when they translate it. So ribosomes read 3 nucleotides at a time, and mRNA has the nucleotides just like DNA does. And these are known as codons. 3 of these together are codons, and this is how the ribosome reads it. Now there are 64 different codons, and they are specific to what they code for. There's only one codon that will tell a ribosome to start building a protein, using an amino acid, and this is AUG. You must have this in order to start building a protein, or else it will not start. There are 3 codons that will tell a ribosome to stop building a protein, and these are UAA, UAG, and UGA. If this is in the code, the mRNA will fall off of the ribosome and it will no longer read it. We will no longer have any more amino acids come in. Each codon codes for a specific amino acid. So it's not whatever amino acid it feels like bringing in. It is specific to the amino acid based on the codon that it has and the nucleotides that are being read. So the sequence AUG, AAC, GGU would be read AUG, which remember is the start one. So the ribosome would then say, okay, I have to start. And then it would read AAC together, and then GGU. And the AAC and the GGU would code for specific codon, or are codons that would code for a specific amino acid. Now, this is your codon chart. If we look at this very carefully, you're going to know this and love this. So, how do we use this? First off, your first letter in your codon, you're going to look at the middle of the circle. Then you're going to look only at that quadrant. So let's say you have the codon UCC, okay? So we're going to look here and we're going to find the U right here. And then, so we're only going to look at this quadrant here because we know our first letter is the U. Then we're going to look even closer, and this is where we look for our second letter right through here. And we're going to look at that and we're going to say, oh, okay, I'm going to look at these and I want UCC. Well, this was U, our first letter. Here's C, so we're going to look at this one. Now, we're going to look only at this section for our third letter. And for our third letter, we're going to look right here, UCC. And as we can see, that codes for the amino acid that is called serine. So UCC, the codon, would code for the amino acid serine. So go ahead and do the transcription and translation in your notebook. And we're going to move on. So how do the amino acids get to the ribosome? Well, a molecule called tRNA carries the amino acid to the mRNA at the ribosome. The T stands for transfer, as the RNA molecule transfers the amino acid. Now, the tRNA also has 3 nucleotides, and it is still an RNA molecule, which are complementary to the mRNA strand being read. So it's going to come in, and these nucleotides are known as the anticodon. So if the mRNA strand that is at the ribosome being read has the codons AGG, then the tRNA that comes in would have the anticodon UCC, and it would have the proper amino acid attached. And then for the next one that's read, on the mRNA, the UCA, the next tRNA that would come in would have the anticodon AGU, and so on. So the tRNA is like a taxi for the amino acid, and once the amino acid is dropped off, recall that it bonds using a peptide bond in between the amino acid. So this is just an overall picture of transcription and translation. In the nucleus here, you have the DNA that becomes the mRNA that's going to leave the nucleus, and then it's going to go out to the ribosome. The ribosome is then going to read your mRNA, which is then going to code for a tRNA to come out, which is then going to bring in your amino acid, which is going to bond the amino acids together using a peptide bond. And this is how we create all of our proteins. Okay, I'm going to see you in class, and we're going to review this even more. I know this is a very heavy topic, so just kind of review it a little bit, and we will talk about it in class a little further.